So good afternoon, everyone. As Esther already introduced, we are located in Munich, specifically at the Technical University. And some of you might know, we have two large universities. One is the Ludwigs-Maximilians-Universität, which also has a medical faculty. And we are situated here at the medical faculty of the Technical University. Some people don't know that you can also study medicine in a technical university, so which has some advantages, I have to say, because it opens up, of course, especially at a later stage in your career, the possibility to interact with different kinds of disciplines, especially the technical disciplines. And this is why you will see in a moment that one of the topics of my group is actually called Novel Technologies in Global Health. So the group's name used to be Parasite Immunology, Esther. It has changed in the past 2 years and has geared up towards infection and immunity in global health, because we align ourselves to the global health agenda, obviously. You have nicely introduced my profile. I don't need to do that yet again, just in case one or the other should be interested. Please do visit our homepage. We're extremely proud of our new homepage that my team and we actually generated together for our group to not only flag publications, but actually to give you the possibility to ask questions, and we try to anticipate questions that you might have for the different topics that we address within our research already upfront when you visit the homepage. So let's start, and let's start where we come from and what we believe in and how we actually transitioned in the past few years. And this overview in the next 25-30 minutes should be rather giving you brief snapshots of what the group is been doing and why we align ourselves to the global health agenda, and where we come from actually. And we come from basic science / translational science, because we're here at the medical faculty. And actually we started the group with a lot of in vitro and experimental model work, some of which I will showcase, not everything for sure. But then since we've been working a little bit in isolation, I have to say, because we were working in NTDs, neglected tropical disease. So we were working in schistosomiasis, and as you can imagine, schistosomiasis, big groups in the US, big groups in the UK, but there was not a lot of attention for neglected tropical disease 10 years ago. So this is why we kept on working and concentrated a lot on understanding host parasite immunology, aligning ourselves a lot with the basic immunology, and asked questions there, try to come up with solutions, but not being really translational. But that then started to develop once global health sort of moved a little bit more to the forefront and agendas were put forward, especially the 2030 agenda. And now the group is trying to really cover and bring in what we've learned from basic research into translation, but aligning at the same time aligning ourselves with SDG 3, especially the targets within SDG 3s in the end to improve health and well-being. So this cannot be done in isolation, as I said before, and we strongly believe, because we're a big wet lab, that global health already starts here in Munich at our institute, and that means that our people that work here, our students, our PhDs, our postdocs, are really international. So we try to really build capacity already here from the very start for students coming from different, also low and middle income countries on the site, capacity which they can take back home to build their own research groups, for example, and to continue their research. So global health goes local, obviously. You have already heard a very nice lecture by Daniela Fusco about neglected tropical diseases. That's why I think I do not need to introduce them in detail to you. This is just an overview slide. But simply to highlight that it's a diverse set of mainly infectious diseases, actually, except for snake bite envenoming, 20 diseases per se, but they have a few common traits, but most important, they are devastating in terms most of them cause chronic disease, disability, and they are the diseases of the poorest of the poor. So these are the communities that need to get more attention, and this is why Peter Hotez has termed or collected these diseases under this term neglected tropical diseases, which was obviously anybody who's working in that space is really groundbreaking change, and was a game changer in my eyes. To really bring those diseases forward, put more attention also from policymakers into developing, for example, new tools to combat these diseases. So our group has started to work specifically on schistosomiasis. So we have the complete life cycle of schistosomiasis established in our institute, in my lab. It's a quite complex life cycle. I introduce it to you in a moment, and believe me, it's true biology. So this life cycle is not always very predictable. So snails do what they want sometimes during winter times. They are not easy to breed. Mice also are not, even though they're maybe genetic clones, infection can run very diverse. So that's keeping up this life cycle per se is a challenge, but it's still fun to work with it. And bilharzia is actually one of the most important neglected diseases, if not the most important neglected tropical disease. WHO currently calculates approximately 203 million people that are being infected worldwide. This is almost as much as malaria. There are 3 major species, and I'm just mentioning this not for you to confuse you, but they cause different clinical symptoms. One is intestinal, the other would be orogenital. I'm sorry, intestinal is really Mansonoid and Japonicum, hematobium urogenital. So this is correct, but they have a diverse set of hosts, and Mansonoid and hematobium humans are the main host of the disease. But in Japonicum, hosts are very diverse, and actually Japonicum is actually considered a zoonotic disease. It's a waterborne, all of them are waterborne, but Japonicum is the most zoonotic one. Like I already said, determinants of infection tend to be poverty, inadequate sanitation, and proximity to livestock, especially with those zoonotic diseases. Thus, tackling those diseases, of course, warrants that you think about them in a more holistic manner, and thus, for example, apply a One Health approach once you think about eliminating a disease. Because you have to think about the host, but you have to also think about the vectors, for example. So, then there are also a few special and very neglected areas of schistosomiasis. One of them I want to briefly mention is the so-called female genital schistosomiasis, which we have considered as important also in our work, and thus have taken this subject on board. The life cycle, as I mentioned, is complex, and again, I don't want to be too technical. Just remember that this is a life cycle which, I hope you can see my mouse, circulates between an intermediate host, which is a freshwater snail, and the so-called definite host, which is either humans in the case of Mansonoid, or, as you can see here, mammalian species like cattle in the case of Schistosoma japonicum. But what is emerging in the field in recent years are so-called hybrid strains. So there are animal schistosomes that actually interlink with human schistosomes and then become hybrid strains. And also we still don't know a lot about these hybrid strains, but we know at least that those hybrid strains, they can travel with people, with migrants, for example, and then they end up, for example, in Corsica. So in Corsica, since a couple of years, schistosomiasis in the southern region is established. So WHO has come forward, and I mentioned this already, with a roadmap to eliminate schistosomiasis. And this roadmap actually has three main pillars, which are to develop novel diagnostics, novel drugs, and to implement targeted control via so-called WASH, that's water, sanitation, and hygiene. And overarching, it is clear that it is necessary to have integrated approaches to work across sectors, so also with the implementation sector, for example, to eventually reach universal health coverage and especially to enforce country ownership. And I will show you in a minute how we try to envisage this. We collaborate with quite a few countries, obviously, in sub-Saharan Africa, in regions where not only schistosomiasis but other NTDs are very prevalent. And together with these collaborators, we try to investigate a few aspects of schistosomiasis drugs. So how do we do that, and what do we want? So coming now to those three topics, I'll give you a few examples. Our group in general has four major topics, and I will start with the first one, which is novel technologies in global health. Why do we need them? And this is just an example. So on the roadmap, since we are employing more and more treatment strategies like mass drug administration in concerted programs, we are moving towards reduction, but also towards elimination of disease, right? This is the ultimate goal. But how do you monitor elimination? You need very sensitive diagnostics, and most of the diagnostics that you use in the field these days are not very sensitive. So we need novel diagnostics, but we also need novel technologies. And this is just one of the projects where we are developing a new PCR for schistosomiasis to detect multiple species based on a novel platform technology developed by physicists from the LMU, which have founded a startup company, which have developed this tool that you can see here. It's a small tool, it's field applicable, you can take it to the field. And eventually this will hopefully help us to fill some gaps in diagnostics, which are to test in the field. Are the snails still infected? That's called xenomonitoring, for example. But also to test, which is quite still not consensus testing, for example, for female genital schistosomiasis. And of course, this cannot be done in isolation. So this is a project by a postdoc driven forward within the DZIF, the German Center for Infection Research, where we interact within our thematic unit, malaria and NTDs, with other researchers, especially in the implementation research field. So jumping from FGS and diagnostics, it's obviously also important to investigate what are the real challenges on site. And here we collaborate with partners obviously where this kind of disease is prevalent. And this is just an example of Gabon, of CERMEL, Centre de la Recherche Médicale in Lambaréné in Gabon, that some of you know, where we have a DFG funded project up and running. But within that project we also performed a very nice interdisciplinary multi-stakeholder workshop exactly to identify the gaps that we need to combat schistosomiasis exactly on site. And this ranges from knowledge, generating knowledge and awareness. Actually, half of the participants of this workshop were midwives, and very interestingly, those midwives, even though they know schistosomiasis, they didn't know FGS, for example, and they were really intrigued by this. And they were also intrigued, as you can see here. This is Amaya Bustindui demonstrating to the midwives a field applicable gynoscope, right? So these kind of tools will help to diagnose and to eventually also eliminate. Now, drugs and drug development. What do we do in drug development? As I told you, Schistosomiasis is a complex disease. It requires a host, and obviously if you have the life cycle established in the lab, you don't use humans as hosts, but you use mice. So we can infect mice and thereby keep up the life cycle. But how do we now test novel drugs? Just assume you were to do the drug repurposing studies, right? So old drugs for new indications like we did in COVID-19. That's, of course, you want to avoid using too much mice, don't even talk about humans. So would be nice to be able to test drugs in vitro, and there are already in vitro testing platforms. But remember the life cycle, you saw it was very complex. It's also very complex because it entails different developmental stages even. So it's not only two hosts, but different developmental stages. This also means that drugs, for example, we have only one drug currently available, that's praziquantel, and praziquantel actually only targets adult worms. So it does not target migrating larval stages through the skin, through the lung. So, to make a long story short, what we have recently developed is a system how we can raise skin stage, lung stage, early liver stage, and even, as you can see here on the lower right side, little worms in vitro without using full blood. So up to now, this was only possible by using full drug. We have now a new system, and this system allows us now to test those different life cycle stages and in vitro, to develop them in vitro, and to test new drugs and how their reactivity. This is in concordance with the so-called 3R principle, which is becoming more and more relevant to avoid animal experiments. So we want to replace animal experiments and also to refine them eventually. And as you can see here, this is just an example of what we do. So we have recently screened a drug library from the NIH, provided from the NIH, and as in the upper gray schemes, you can see these are on the number 3, these are really healthy live worms. 0 is obviously they're all dead, so there's a scoring system that you can apply. It's a visual scoring system, non-automated, very biased towards the observer for sure. So it's not easy, still a lot to develop. But anyway, as you can see here on those graphs, in yellow you have praziquantel, so the viability goes nicely down, and we have identified 2 new drugs which could be developed further into potential drug repurposed drugs. We have also made a nice observation during our studies, and this is an observation that MSE, as you can see here, is on the scale. You see the different developmental forms of worms in vitro, and with human serum they develop really nicely. But if we add mouse serum actually kills those schistosomes in vitro, which is actually per se already quite intriguing. But our student here, Johanna, and our postdoc Fabien, they're currently together analyzing using high throughput screenings what in mouse serum actually kills those schistosomes, because that again could identify new targets in schistosomes to develop new drugs. Now coming to one of the core topics, which are the developmental origins of health and disease. So what is that? Developmental origins of health and disease is actually it has been put forward already many years ago, stating that in utero and early life experiences can actually determine the propensity to develop non-communicable diseases later in life. So this has been put forward, for example, in population studies where populations that have been exposed to trauma such as war have born children which during adult age suffered significantly more from heart attacks, severe heart attacks. This is just an example to give you you, from these old studies, the theory of developmental origins of health and disease actually is derived. Environmental conditions of early life are linked to state of health and risk of disease later in childhood and adult life. However, this kind of research is almost exclusively has been performed in high-income countries. Not a lot is known, and only very recently, you can see here, people have started to take up this discussion. What about low-income countries? And along these lines, especially considering that we have now that health also in low and middle income countries is in the end changing, and we are seeing more and more non-communicable diseases in these countries. Of course, this developmental origins of health and disease theory could play a very prominent role here, but maybe it already has without us knowing it, because now, coming back again actually to schistosomiasis, we know that 40 million women at childbearing age are actually infected with schistosomiasis and chronically infected. And you might remember Daniela is also working in that space. So, but what we are interested in now is to ask ourselves, what is now the relation of chronic infection during pregnancy and allergic disease as a hallmark of non-communicable diseases later in life? And we have done this with a lot of mouse models, and I will not go into this much deeper, but just to keep in mind, for you to keep in mind that helminths per se, why do we come to that theory, are master regulators of our immune systems, and they have bystander effects on non-communicable diseases and vaccines. So this is just to summarize a lot of work, and I will just guide you very briefly through this experimental setup so you grasp the background completely. So what we have done, schistosomiasis is for immunologists. Schistosomiasis is really intriguing because schistosomiasis in the end has 3 very distinct phases. So, and if you made now female mice that are infected with schistosomes during those 3 phases and generate offspring, and now you subject those adult offsprings to allergic airway inflammation model, you find suddenly that those that are long-term, where the mothers have been long-term infected, are protected against these allergies. So this could have implications, of course, for humans. And I will just wrap up. We have an ongoing study, the so-called helminths study, where we're just really trying to understand how this fetal-maternal interaction during schistosomiasis actually imprints on many surrogate markers of the immune system. I come to my last topic, which are the NTDs and translational immunology, and give you the example of another neglected tropical disease, which is cysticercosis or neurocysticercosis, which is a zoonotic disease which circulates between pigs and humans. I will not go into detail, but we are working with, again, not in isolation, but with partners from 3 sub-Saharan African countries, really in a multidisciplinary network which places research, capacity building, networking, just as in helminths, especially in partnership, at the forefront. And what we as a group are investigating is a very central question, is the finding that viable brain cysts mostly remain asymptomatic, whereas cysts that are dead or die due to therapy mostly cause symptom onset. So there's 2 theories out there. Could it be parasite specific, so due to those different developmental stages, or due to the host immune system reactivity? And in a recent review, we've summarized the open questions, and I just highlight here, you just have to see there are a lot of question marks obviously here pertaining to the parasite itself, so the viable or the dying cyst, and the reactivity of the immune system. And recently Fabien has published a really nice paper where we have identified and answered a lot of these questions, and where we are now moving towards the translation. So what we learned, in the end, we have learned that indeed those different cyst types, so viable versus dying cysts, release very different kinds of products which stimulate the immune system in a very differential manner. And now we can use this information to develop biomarkers for diagnosis of stage of the disease, as well as guide patient treatment. Is that a patient that reacts massively with a lot of inflammation? Yes or no? Now, coming to the final slides on GLOHRA project, and you have now, up to now, all gathered that helminths in general, so NTDs in general, are chronic diseases. They cause, therefore, bystander effects because they are big immunomodulators. So they not only modulate allergies, but they also modulate vaccine efficacy. And this has been demonstrated in mouse models quite extensively. We have also contributed to that field. And now the big question is what obviously happens in humans, because that has a lot of implication, right? Chronic infection, the burden of disease is as high as 70% in some areas of any kind of NTD or chronic disease. So it is relevant to look into the literature. What do we actually know about humans? And this is what all this new study is about, the Helm-CIS study, as we call it, which is a systematic review which comprehensively analyzes the impact of helminth infection on vaccine performance in humans. And of course, we have developed in the past weeks and months a study protocol together with our colleagues, Miral Esen, Stefanie Klug from the epidemiology department, and Miral Esen from the clinical studies department at the University of Tübingen, or the Tropical Medicine Institute. And this is spearheaded by Mariam Habib, who's the major PhD in this project. And where are we at the moment? We, of course, there is a timeline that we have put forward, and study protocol has been developed now. We have published this in Prospero. We're now on our search and screen, and a lot of discussions going on to assess the quality then eventually of these publications, to write up hopefully very nice manuscript. So what is the impact of that in the future, or could be the impact in the future, is of course then the design of large-scale clinical trials to really systematically investigate the impact of helminths on now ongoing, ideally on ongoing COVID vaccine programs, and to overhaul mass drug administration. So meaning, could it be that treatment of helminths could actually enforce vaccine efficacy at one point? So when do we vaccinate? When do we treat? So, of course, also my group, we don't work in isolation, and as mentioned before, I'm the co-founder together with Andrea Winkler, who you see here, of the Center for Global Health, and this is just to showcase what we have done during COVID-19. Of course, that's the year 2020 when Global Health went local, and also for us that meant being here, not being able to travel. That meant webinars, presentations, and this is of course lectures, and this is of course what we did. And with this, I'm already a little bit over my time, 2 minutes, I'm sorry for that. I would like to close and just close with this. Global health research is an area of study, research and practice that places a priority on improving health and achieving equity in health for all people worldwide. And I think that's still a very nice definition. It's quite old, but it still holds true. These are our partners, these are our funders, just to acknowledge them. GLOHRA is one of them, as you have just seen, and I'm really happy to take questions, but not with acknowledging again the whole group. This is a picture taken recently. Of course, there is fluctuations as always in research, but I thank you very much for your attention.